Hello, everyone, and welcome to the webinar. So my name is Sofia, and I'm a colleague to Urmas in communications. I'm working in communications, and Urmas is sitting right here beside me, ready to get started. Urmas, you will be presenting for approximately 20-25 minutes, and then we'll turn to questions. And after that, we'll open for questions. So if you have any question, please be sure to write it down and send it through to me via the chat function in the meeting here. And then I will pass your question on to Urmas after the presentation. But Urmas, over to you.
Thank you, Sofia. So welcome to this webinar on 3D imaging. So my name is Urmas Roostalu. I'm the manager of the 3D imaging department at Cobra. In our department, we use and offer 3D light sheet imaging as a service for our contract research customers, but also use it in our internal development projects. So we are always working on developing new imaging methods to make it possible to image more diverse tissue types, but also to establish new endpoints for preclinical research and drug discovery. So today, in this brief webinar, I will go through a few topics related to light sheet imaging. So first of all, I will very briefly introduce you to the basic principles of light sheet microscopy. Then I will discuss how to prepare samples for 3D imaging. I will show you also some examples of light sheet imaging in different organs. And eventually, I will also discuss some of the challenges that we have faced, and maybe some of you also are facing in 3D light sheet microscopy, and how to address these to design perfect imaging studies. So light sheet microscopy, in very simple terms, relies on moving the sample, so like a heart here in this schematic, through a laser beam. And a camera that is located either below or above the sample is then actually capturing images at every 2 or 20 micrometer or whatever interval. So and this really depends on the research question. The light sheet microscopy is made possible by tissue clearing. And here in this movie that is playing now on the left, you can see a mouse brain becoming transparent in front of your eyes in a matter of half an hour, 1 hour. So what happens here is actually the brain is put inside an organic solvent that we call ECI, so ethylcinamate. And what is happening is actually refractive index matching. So you match the refractive index of the organ with the solution around it. And by doing this, it actually becomes possible to see through the tissue and visualize the fluorescent signal that is inside it. So for instance, here you can see a mouse brain, where you can identify all the large blood vessels by standing for vascular smooth muscle marker. And because refractive index matching, the transparency of the tissue, you can see clearly deep into the sample and identify all the vascular structures. Light sheet microscopy and tissue clearing is not only possible for smaller samples, but also for those significantly larger. So for instance, here is an example from a rat brain that has been made optically transparent and stained afterwards or in this process for tyrosine hydroxylase, which is a marker for dopaminergic neurons. So these are the cells that are commonly lost in Parkinson's disease. Moving on, so into the question of how large samples we can image with light sheet microscopy and what is the resolution limit. So the sample size actually is, it can vary a lot. So we image sometimes samples that are just barely a millimeter, but then all the way up to 3 centimeters or more. So there is a balance of what is actually required to answer a specific research question. And the resolution, we can image structures just like 2 to 5 micrometer in size. For instance, here in this images on the left-hand side of the slide, you can see a mouse brain where the capillaries have been visualized by antibody staining. So you can actually track every single capillary in the entire brain and see how and when they are connected. There are nowadays also some technologies that enable to push the resolution limit even further and see subcellular structures. So using, for instance, expansion microscopy. Next, I would like to briefly discuss the possibilities of multiplexing and imaging different wavelengths. So here, in essence, light chip microscopy can image over a number of different wavelengths, starting from green short wavelengths up to near infrared wavelengths. But each of them actually has some advantages and disadvantages that one needs to take into account when designing a successful imaging study. So here below you can see a movie playing where we have visualized arteries, veins, and capillaries using antibody staining in different channels. So when discussing about the wavelengths, then shorter wavelengths like green fluorescent protein wavelengths, Alexa 488, they are perhaps not the best ones for light chip microscopy because light at this wavelength doesn't penetrate very well into the tissue. At the same time, there is also quite a lot of autofluorescence in the background. So I would recommend, if you want to use something in this wavelength, then perhaps pick only the best or very abundant targets. So when we move further into a little bit yellowish red wavelengths like Psi3, Alexa 546 or Alexa 594, then things become already considerably better. So the light penetrates well, but still here you have some background fluorescence in the tissue visible. When we move further into the closer to the infrared and far red spectrum using Psi5, Alexa 647, or furthermore Psi7 and IR800, the things become way better. So this is the sweet spot for light chip microscopy. Light here penetrates very well into the sample, and you have just very little almost non-detectable background signal. So by combining these different wavelengths, we can do these kind of images like shown here for the different vascular markers, but it can also be many other applications for this. So what are the applications for light chip microscopy here? We at Cubra, we use it very often, for instance, for compound biodistribution. We have a drug that is fluorescent. It can be a peptide, an antibody, or something completely different. And if it is fluorescently conjugated, then we can answer a question where the drug end up in the mouse, where it has been dosed, either, let's say, into the circulation IV or into the muscle. And then we can see how much it actually reaches the desired organ, for instance, the brain. The second question that we commonly answer is drug target visualization. So, for instance, looking at where a receptor is localized, in which cell types, and this can actually complement the first question of compound biodistribution. We use it frequently also in characterizing the pathology of the in a disease model, for instance, looking at vascular rarefaction, fibrosis, Alzheimer's plaque formation, inflammation, and so on, and then seeing if a compound has an effect on these different endpoints. And last but not least, we very, very commonly use it for neuronal activation. So how much a drug leads to neuronal activation in the brain, looking at early activation markers like CFOS and BSTAT3. In the next few slides, I will discuss a little bit how to prepare samples for these different experiments. So perhaps the simplest experiment is actually a compound biodistribution experiment. So here you can see a movie where peptides, so this is GLP-1 receptor agonist, has been conjugated to a Psi5 dye. And this is a small fluorophore, and you can very nicely see the accumulation of the dye in the brain where the compound has reached the tissue. And this is in a way easy because the mouse has been dosed with a compound, then it is perfusion fixed with neutral buffered formalin and overnight fixed, and then afterwards the sample is stored in PBS until we are ready for imaging. It is important to avoid freezing, which may damage the tissue, and it is important to avoid any kind of bleaching, which may diminish the compound signal. So the sample is gradually dehydrated in methanol and stored in ECI for imaging, and afterwards we can use various image analysis algorithms to quantify the signal in the sample. When we move on to immunohistochemistry in 3D, it becomes maybe a little bit more challenging. So here you can see a movie where we visualize tyrosine hydroxylase staining in a mouse brain. So you can see the dopaminergic neurons in the substantial library area here in the middle. So in essence, the start of the protocol is somewhat similar to a perfusion, fix a tissue by intracardiac perfusion. The fixative though can vary. So we have evidence that some antibodies work better with glyoxal, others in PFA or NBF fixative. So sometimes it may require some optimization here at this stage. And afterwards, we store the sample in PBS if we can start in a relatively short time period, and then use different immunohistochemistry protocols like published protocols, IDISCO, ADIPO-CLAIR, Chanel, depending on the tissue type of interest. And this kind of signal can also be quantified. And finally, I will briefly discuss mRNA imaging, so 3D in situ hybridization. So here you can see a movie of a mouse brain where we visualize 3 different mRNA targets: parvalbumin in blue, tyrosine hydroxylase in yellow, and somatostatin in red. And in this case, we fix the mouse by perfusion with paraformaldehyde. We store the sample in methanol at -20 degrees Celsius, and then use hybridization chain reaction technology to visualize the transcripts in the brain, and then quantify it afterwards. So these are just some very general guidelines, and maybe just a few remarks on the different organs and tissue types. So for the brain, it's good to avoid any kind of tissue damage that may lead to problems afterwards in image analysis. For blood vessels, it is important to eliminate blood very efficiently by perfusion, because this may lead to autofluorescence and issues. For lungs, it is necessary to perfuse them via the trachea and not IV via the circulation to maintain the morphology. And for the hearts, we actually prefer to stop them in diastole before fixing to enable better comparison between the samples. So now in the next section, a few slides, I will actually show you some of the examples from different organs and how they look like and what can be done. So first of all, let's move to the brain again. So this is a mouse brain stain for a neurofilament antibody, and I have to say that brain is the immunohistochemistry and 3D imaging actually works very, very well, and often requires little optimization. And most antibodies work either primary and conjugate, secondary sequentially, or primary antibodies conjugated. And IDISCO protocol or similar protocols usually enable very nice staining. And what makes brain imaging particularly appealing is actually the capability of quantifying signal across all the different brain regions. So mouse brain has this atlas that has region coordinates and volumes for more than 400 brain regions. And at Cobra, we often use this drug-induced CFOS imaging. So you can see this in a movie here, a mouse that has been dosed with GLP-1 receptor agonist, semaglutide. His brain has been isolated and stayed for CFOS. And every dot that we see here in this movie is a neuron or a cell in a mouse brain that has upregulated CFOS. And this enables us now to transfer this data into a mouse atlas framework that you can see now on the images on the right. So we can actually take several brains in a treatment group together, for instance, in this experiment, 6 of them, and then make the average response map and compare it to a vehicle dose control group. So in these brains, we can actually, in the middle panel, you can see mRNA level quantification, and on the right-hand side, protein level quantification of CFOS positive cells in a number of brain regions. So brain regions that in these average brain maps that are red have a higher CFOS cell count compared to vehicle. And these charts with these colorful red, blue boxes highlight a few brain regions out of these hundreds that have a known function in appetite and satiety regulation. And where you see these dashed lines and bold borders, these are the brain regions that are significantly different between vehicle-treated and semaglutide-treated mice. So this kind of brain imaging actually enables to identify which brain regions are activated. Are these brain regions the ones that we desire to activate, that have a known function in a given disease area, and also identify unwanted side effects in the brain. So next, let's move on to kidney imaging, which is in essence actually relatively similar to brain imaging. So here on the left-hand side, you can see a mouse kidney stained for KIM1, which is an injury molecule upregulated by epithelial cells in the kidney tubules in a kidney injury mouse model. And in magenta, in the same animal that you can see in the same movie in a moment, you can see podocytes, so the glomeruli labeled as well. So this kind of imaging is relatively similar to the brain. You can use IDISC or antibody staining and so on. Sometimes, however, maybe you don't need antibody staining at all. And this is, for instance, evident now on the movie on the right-hand side. So here is a kidney from a polycystic kidney disease rat model. And here we just image autofluorescence at the lower wavelengths. And here you can actually, just based on the autofluorescence itself, you can see these large cysts that are forming in the kidney. And this provides very nice morphological information of cyst formation, and we can use it to characterize if a drug has an effect on that cyst volume. Both in the kidneys, but also perhaps in other disease models, you can also look into fibrosis, like looking into collagen. So here you can see a kidney that is a healthy mouse where collagen is primarily present in the walls of large blood vessels and a little bit in the renal capsule around the kidney. On the right-hand side now you can see a mouse heart. So this mouse comes from diabetic and hypertensive heart failure model. And in this case, you can actually start to see perivascular fibrosis, but also interstitial fibrosis developing in the sample. And this imaging can help. Now you can see in the middle of the heart these large fibrotic patches to actually characterize the volume of fibrosis and make then conclusions based on the effect of fibrosis and on heart function in general. Imaging is possible actually in almost, I would say, actually all mouse organs. Some may require some special attention and treatment. For instance, here you can see a mouse heart, mouse eye, and in the eye there is a requirement often if it's from a pigmented mouse to bleach out the dark pigment, so you can actually see the retinal vasculature behind in the back of the eye. Some other organs have different requirements. For the bones, they need to be decalcified to enable antibody penetration and tissue clearing. And some of the internal organs, like a liver and pancreas, may benefit from prolonged delipidation and dehydration to enable nice clearing. So here you can see a mouse pancreas, where nice blood vessels are staying in green, or this vascular smooth muscle marker. And now you can see insulin-producing beta cells in the islets in this glow scale in red. And in this kind of experiment, you can actually visualize every single islet in the entire pancreas and characterize how well the drug affects islets in diabetes mouse model, for example. So I have so far discussed mouse and a little bit rat imaging, but actually light imaging is possible also from tissue biopsies from larger animal models or even from patients. And in this case, of course, you can have the fusion fixation, but fortunately enough, light imaging also works when a sample is immersion fixed in formalin, for instance. So here you can see a movie. So this is a biopsy from a big atrium where tyrosine hydroxylase positive neurons have been visualized by immunohistochemistry. So in this kind of experiments, there may be sometimes a requirement to use some specialized protocols that loosen up the extracellular matrix or enable better antibody penetration. For example, Charel and Summit that have been published in the recent few years. So in the next part, I would like to address some of the challenges that one may face in light sheet microscopy and how to address these. What are the common problems? So what we have sometimes faced these problems due to sample fixation. So most of the rodent studies, they require or they greatly benefit from diffusion fixation. However, wrong diffusion fixation pressure, for instance, can lead to issues. So if the pressure is too high, the capillaries may start to break and leak, and then of course it has a negative impact on tissue morphology. If the pressure is too low, on the other hand, you have some blood left in the tissue, which is out of fluorescent, and that doesn't result in nice images. I have already previously mentioned freezing, but actually I bring it out here as well, because sample freezing, if it is in the aqueous solution, it leads to ice crystal formation in the tissue, which then when it's eventually thawed up at room temperature, causes damage in the sample and also has a negative impact on morphology. So of course, one has to be careful with tissue dissection as well. And now, finally, when we're coming to the lab part, what some of you who is also hands-on developing imaging in the lab may face is insufficient antibody penetration into the sample, for example. And there are few ways how we address it at Cobra. So one of the examples is shown here on the right. So we have found that using conjugated antibodies can sometimes offer a nice advantage and actually solve this issue. So on the upper panel here, you can see a whole mouse brain that has been stained with a vascular marker CD31, and then with a secondary antibody recognizing it. And this image is a plane, 2D plane from a 3D image brain. And you can see that in the box number 1, so the uppermost one, you have a very nice vascular signal in the capillaries, but the deeper you go to box number 2 and 3, you see less and less blood vessels. In the lower panel, you can see the very same antibody, but then conjugated to fluorophore. And there you can actually see that the signal in the different boxes, 1, 2, and 3, is quite similar indeed. So antibody conjugation can actually help you to solve some of these issues and get a nice signal. And there are, of course, some other possibilities that one can take. For instance, changing the antibody concentration, using more or different detergents to enable better penetration, or just incubating longer. And, this usually helps to achieve nice imaging results. So last, just a few final points on how to then design a successful imaging study. So if it's a completely new endpoint, new antibody, mRNA target, then perhaps it's sometimes useful to carry out a small pilot experiment before starting with hundreds of samples in the same go. If it's a qualitative study, then sometimes just N=3 is enough per group to get an overview of where particular signal or cells are localized. But if it is a quantitative study, then most often we have a group of around 8-10 animals per group, per treatment group. But this, of course, depends on the disease model and if there are previous power calculations and so on. So, and then related to this little bit is also like the controls that are needed. So which kind of vehicle is used as a control and how to correct for autofluorescence, for example. And finally, they come a little bit hand in hand. It's the resolution that is needed to answer a particular question, preclinical research, and how to handle data. And of course, it is nice to see every detail in the sample, but this comes at the cost of data. So at Cobra, when we image just one single mouse brain, we typically end up with 300 gigabytes of data. And even if we downsample it into more manageable format, it still is 20, 30 gigabytes per one single sample. And this adds up to a large data amount over over hundreds of samples that we continuously keep on imaging every week. So this of course has some implications on the IT infrastructure, and we are lucky to have a very good server and infrastructure here at place that enables us to store and actually analyze these large data amounts. But this is just something to keep in mind when starting up these experiments, that do you actually have what it takes to store and analyze the data. So I think with this I am actually finished with my webinar now. So if you have detailed technical questions, you are very welcome to send me an email in this email address you can see on the screen now. But more general questions I'm actually happy to take now.